Welcome to the program. Lessons for Gene Drive from Ecology and Invasive Species Management. This is a program produced by the Gene Convene Global Collaborative. I'm Dave O'Brockton, and I'm going to be your host and moderator for the program. Let me begin by saying a little bit about the Gene Convene Global Collaborative. Gene Convene is a program within the Foundation for the National Institutes of Health that focuses on the application of gene drive technologies to pressing problems in public health. Its overarching mission is to support informed, responsible decision-making. It does this by partnering and stakeholders to identify and address key questions, provide technical advice, strengthen capacity, share information. You can learn more about the Gene Convene by visiting their website at geneconvene.org. Let me continue by just giving you a little bit of background that's relevant to today's program. Gene drive technologies are genetic constructs that are preferentially transmitted from generation to generation. When a gene drive construct is introduced into a population of organisms, it can rapidly increase in frequency within that population over the course of relatively few generations. These technologies have transformative potential in the field of malaria control and elimination, particularly in Africa. Gene drive constructs can be assembled today that are highly effective and efficient in the laboratory. As yet, there have been no field tests of lab-assembled gene drives. However, developers envision a gene drive containing mosquito being ready for field testing in about 5 years. There remain a number of outstanding questions regarding these technologies, one of those being how the effects of gene drive containing organisms in the environment will be monitored. There are many examples of challenging ecological and biological monitoring situations. Which of these might represent good analogs or orthologs that could be useful to gene drive developers? This program was produced by Gene Convene to begin to explore that question and is part of an ongoing effort to foster thinking and discussion in the gene drive research and development community as it moves towards initial small-scale field releases. We have 4 speakers today. I'd like to briefly introduce them now. Paul Abram is a research scientist and an expert on biological control of insect pests, and he works at the Agassiz Research and Development Center in Agassiz, British Columbia. Paul has an interest in basic biology and ecology of parasitoids, and he develops biological control programs for invasive agricultural pests, including natural enemies. He also has an interest in assessing the ecological risks and benefits of accidental introductions of natural enemies that attack crop pests. Biological control involving widespread release and establishment of non-native insects might be a useful analog for aspects of gene drive-based biological control. Paul will focus today on monitoring and surveillance of introduced biological control agents. Renee Bellinger is a research geneticist at the U.S. Geological Survey's Pacific Island Ecosystems Research Center, which is based in Hawaii Volcanoes National Park. Her research focuses on, among other things, applying genetic tools to understand population dynamics of native and invasive species. He's involved in a project aimed at mitigating the impact of an invasive mosquito species that transmit bird malaria, a disease that threatens a number of indigenous bird species in Hawaii. The control and suppression program that aimed at these mosquito species is based on a genetic biocontrol strategy referred to as the incompatible insect technique. She'll tell us about some of the challenges faced in bringing this control strategy to the field, including those around monitoring and surveillance. Jafet Asunu is a lecturer in statistics and data analysis in the University of Glasgow School of Mathematics and Statistics. He's interested in developing statistical models to analyze biodiversity data from citizen science projects and applying Bayesian methods to characterize biological communities in changing environments. One of his current projects looks at methods to estimate species distribution while accounting for observational process of how the data are collected, across different citizen science biodiversity monitoring schemes. Finally, Sarah Paul is research scientist in disease ecology at the National Ecological Observatory Network, which is headquartered in Boulder, Colorado. Sarah is a disease ecologist whose research addresses questions at the interface of ecology and public health. She's broadly interested in studying how interactions between humans, animals, and the environment influence infectious disease risk. Among other of her responsibilities at the observatory, she oversees the data associated with small mammal, mosquito, and tick communities, along with the associated pathogen data sets. Sarah's going to tell us about the observatory network and a particular data package produced by the observatory network concerning mosquitoes and mosquito pathogens. Finally, let me just give you, the audience, a little bit of orientation. Your cameras and microphones will be disabled during the presentations. Each presentation will be about 30 minutes in length. We're going to take questions and comments after the last presenter. You can ask your questions either by using the chat function, or you can raise your hand and we'll unmute your mic. You should feel free to chat in your questions at any time during the presentations, and we'll take them at the end of the last presentation, as I mentioned, in the order that they're received, or I may mix them up and take them by different categories. In any case, I encourage you to ask questions at any time. This program is being recorded and it will be posted on the Gene Convene YouTube channel at the URL you see on the screen. So now let's begin our program. Paul, I'm going to turn the floor over to you. Thanks for being here. And the floor is yours.
Thanks very much, David. Can you see my screen and hear me all right there?
And yes, I can. Your presentation is up. I can hear you just fine. Thanks again for being here, and take it away.
Thanks very much, David, for the introduction. Today, as David mentioned, I'm going to talk about importation biological control of insect and weed pests, because I think it might be an informative precedent in some ways for gene drive technology. I'm not going to editorialize too much during the course of the presentation about how the lessons from biological control might apply to gene drives, but I hope that everybody listening will sort of think in the back of their mind as I'm presenting about what lessons from biological control apply and which ones maybe do not. So many of you are probably familiar with the basic principles of importation, or what's sometimes called classical biological control, but I want to give a brief overview so that we're on the same page. So in short, the goal of classical biological control is to reunite invasive pests with their natural enemies in the invaded range. So something we often see is that when pests are accidentally moved from their native range to a new invaded range, they outbreak, reach large densities, and cause big problems for people. And one of the reasons it's hypothesized why this often happens is because they've left behind natural enemies that help suppress their populations in their native range. And so the practice of importation biological control involves collecting these natural enemies, redistributing them to the invaded range, releasing them with the idea that they'll propagate themselves and provide long-term suppression of the pest population. The example I've given here is pictured as the biological control program against cassava mealybug in sub-Saharan Africa, which is probably one of the most successful biological control programs ever initiated. And I think many people have probably heard examples, both certainly the failures and also maybe some of the successes of biological control. But I think most folks don't realize how many of these introductions have happened over the last roughly 130 years. So for biological control agents against insects, which are usually predatory, like this ladybird beetle, or parasitic, like this parasitoid wasp insect, these make up the vast majority of these introductions. There have been more than 6,000 individual introductions of more than 2,300 species of natural enemies against more than 500 pest species in 148 different countries. So this is really a global practice. Here, I've highlighted the countries that do the most insect biological control. Canada, the US, South Africa, Australia, and New Zealand are the most active jurisdictions that conduct biological control nowadays against insects. Depending on how you define success, some level of control ranging from partial control to basically silver bullet complete suppression has been reported for more than 226 species of target insect pests using this practice. Biological control is also practiced against weeds, invasive plants, using insect herbivores. Again, there's been lots and lots of these introductions, more than 1,500 releases, more than 400 species against lots of weed species in more than 90 countries. The countries that practice weed biological control most often are the same ones that practice insect biological control the most often. Weed biological control actually has a little bit higher rates of success than insect biological control. About half of the programs fail, and about half of them succeed in getting some levels of target weed suppression. So why am I speaking to you folks here today? Well, I think it's because importation biological control has some interesting similarities, but also differences to some types of gene drives. They're most similar, I suppose, to the low threshold self-sustaining population suppression gene drives. Those similarities are that both methods involve the release of a self-propagating agent into a new environment. And so, folks who are interested about negative environmental impacts are justifiably concerned about the potential irreversibility of these interventions. And both methods aim to suppress populations of an undesirable species, but could also have negative ecological impacts or unintended consequences. Some key differences, though, are that to keep in mind are that proposed gene drives may target native species, whereas biological control, at least the modern iteration of it, usually targets invasive species. And gene drives involve the release of genetically modified organisms, which biological control does not. But obviously today I'm going to focus on the similarities as they relate to biological control. I'm going to have a very, very brief history and 3 points here of importation biological control, because again, I think a lot of folks might not be interested or aware of the full sort of history of this practice. For the first roughly 100 years of this practice, there was really just a focus on the economic benefits that it could provide. So there was very little regulation of the movement and introduction of these organisms, and essentially no research was done on whether or not they were specific or not to the target pest. Then starting in about the 1980s, there was a realization that some of these past introductions had really negative ecological consequences, the most famous of which, of course, is the introduction, the ill-advised, by today's standards, introduction of the cane toad to Australia. So this sort of brought in an era where really the focus, it was on risks. So there was intense criticism of practices of importation biological control based on a small number of releases, all conducted before the 1970s, where there was demonstrated ecological harm. And this resulted in greatly tightened regulation and a drastically reduced number of introductions. Where we are now is that this criticism, especially from the scientific community, has improved risk assessment science. There's really a push to incorporate more ecological principles in risk assessment and biological control, and then a push by some scientists to update the regulatory structure to focus not only on risks, but also on benefits. So that's just sort of to orient you in the history. I just wanted to show you in this graph, showing the number of introductions of weed biological control agents in different decades over time, how the increase in regulation and the sort of tightening of standards for specificity of biological control agents in the last 2 decades has really reduced the number of introductions that are done in the 5 main jurisdictions that practice biological control. And this is against a background of the situation where the rate of plant and insect invasions has been accelerating. So it's not as if there's fewer targets for biological control. This is really because we're being much more selective about the agents that are introduced. Some reasons for the recent declines. I mentioned increased regulation at both the national and international levels. Most candidate biological control agents are now filtered out by researchers early on in the process because the standards for their specificity are so strict. In our field, there is definitely diminished research capacity and limited funding, especially in academia and especially in North America. It's now mostly, in case you're wondering, it's mostly government scientists and some academics that do the research that support biological control introductions nowadays. And then something that I think gene drive people think about a lot is, as well as public support and knowledge, biological control suffers a little bit from what we call the cane toad problem, which is that the public, if they are aware of biological control, usually thinks of only the most catastrophic failure that happened, over 100 years ago now. Okay, sort of moving towards environmental monitoring, most research focus during the evaluation of candidate biological control agents is now on what we call their non-target effects. The type of non-target effects we think about in biological control might be similar to the ones that one would think about in gene drives as well. We're worried about direct effects of the introductions on native non-target species, direct effects on beneficial or valued exotic species as well. Crop plants often fall into this category. Direct effects on other pests, which might be considered positive, in fact. Potential competition or displacement of other beneficial organisms. Indirect effects and indirect ecological effects, which we'll talk about a little bit later on the same or other trophic levels. So how do we monitor for the consequences of the establishment and spread of these biological control agents after their release? In biological control, we call this post-release monitoring. And this is the term that I'll use for the rest of the presentation. Just briefly coming back to, because this is important to understand some later parts of the presentation, how much post-release monitoring has been done during these different eras of biological control. During this so-called benefits era from the 1800s to the 1980s, essentially there was, it might surprise you to know, there was essentially no post-release monitoring done of non-target impacts, or it focused only on non-target species of economic relevance, like crops. It wasn't really until the 1980s when the lack of post-release monitoring was one of the key criticisms that was leveled at biological control, that the research community really started to do non-target post-release monitoring in a meaningful way, because there were very few cases of demonstrated ecological harm, but the obvious criticism was, well, if you're not looking for them after the release, of course you're not going to find them. So the mostly retrospective post-release monitoring of older biological control releases started during this era. It was really during this era, too, that there was a shift in focus towards species of conservation importance, not only non-target species of economic importance. And now, in the last 15 years or so, post-release monitoring of non-target impacts is becoming much more common. It's really a focus of research now. And it's sometimes, it might surprise you to know, again, requested by regulators. I just wanted to give a brief overview of the requirements for post-release monitoring in different jurisdictions that practice biological control. And surprisingly, post-release monitoring of non-target effects is recommended, but not required or enforced in most jurisdictions that regularly practice biological control. South Africa, Australia, and New Zealand, for example, there's no explicit requirement for post-release monitoring, although plans are usually outlined in applications for releases, and post-release monitoring is now usually done by applicants for releases. For example, New Zealand has some of the best science on post-release monitoring of anywhere in the world that does biological control. In Canada and the US, post-release monitoring plan is a required part of release petitions. Petitions are the documents that undergo scientific and regulatory review before the release of a biological control agent. Reports on post-release monitoring are sometimes on a case-by-case basis requested after releases are done, but there's been no documented cases of sort of enforcement of post-release monitoring plans. Europe, I'm going to focus on Italy here, has the strictest requirements for post-release monitoring plans. Post-release monitoring plans are required in petitions to release, and monitoring is actually enforced through annual follow-up. So in order to get a release permit to do biological control releases in a second year, data on monitoring of non-target impacts is needed from the first year. And this needs to be done for at least 3 years. In North America, which I'm going to focus on as a little bit of an example for what types of things are required during post-release monitoring. It's the guideline. We have harmonized guidelines in North America for Canada, the US, and Mexico that are outlined by the North American Plant Protection Organization regional standards. And they actually outline what the key elements to monitor during post-release monitoring of biological control agents are. The first two items basically relate to the establishment, spread, and impact on the target pest of the biological control agent, so not non-target effects. I'm just going to very briefly mention these because I know this isn't really the focus of the presentation today, but the sort of target effects, there are lots of examples of good long-term monitoring programs to measure the benefits of biological control introductions in terms of their impact on the target. I'm not going to present any examples of these today, but suffice to say that often the types reduced population densities of the target pest is the main thing that's usually measured. And then sometimes there will also be measurements of the resulting benefits, for example, in monetary terms, in terms of benefits to native environments, biodiversity or ecosystem services that can result from biocontrol introductions, and less commonly, but there are examples of social and demographic benefits to humans being measured as a result of biological control introductions. Shifting to non-target impacts, though, which I think is what most folks are interested in today, the recommendations from NAPO ask for information on the impact on selected non-target species for which potential impacts are identified. Data collected should include things like the preference of the biological control agent for the non-target versus target hosts, development, changes in growth, survival, and reproduction. So I'll get to the growth, survival, and reproduction part in a minute, but first I want to talk about the types of non-target impacts that we usually measure. So almost exclusively what I'll call direct non-target impacts. So that's whether the biological control agent attacks and kills related native and non-native insects and not the target invasive insect pest. But insect and plant communities are extremely diverse. It's not easy to study every single species, and there's biological and logistical characteristics of every single potential non-target species that just makes it totally unfeasible to measure every single possible direct non-target impact in the environment. Again, I'm sure this is something that gene drive people think about. How do you evaluate potential non-target impacts when there's so many different species to evaluate? So I just wanted to give examples of, there's been a lot of thought and discussion around this in biological control, and researchers have come up with sort of criteria to help decide what organisms to monitor for potential non-target impacts. Some of the key ones are species that are identified as potentially vulnerable in pre-release lab testing. Biocontrol introductions require a lot of data, usually from containment facilities, about the specificity of biological control agents. Sometimes non-target species are identified as potentially vulnerable during that testing, and so those can be good targets for post-release monitoring. Species that are taxonomically closely related to or occur in the same environment as the target species are often subjects of post-release monitoring. We think about whether non-target species are of economic importance, cultural importance, conservation importance. And then there's more practical considerations as well. You know, for example, whether the non-target species that we're interested in is actually experimentally tractable. Can it be reared or propagated for use in experimental work? Is it common and widespread enough in the field to actually monitor with limited resources? And then it can be useful to think of sort of, I know in this group we're calling them kind of indicator species so in biological control, we think about whether those are ecologically or phylogenetically representative of a broader group of non-target organisms that might be difficult to obtain, but by focusing on sort of that indicator species, we might be able to learn something about that group in general. And there are frameworks for sort of taking those criteria and sort of formalizing them into sort of a ranking system for deciding what non-target species to focus on. So this is just an example, the Pronti scheme, which generates priority ranking values for non-target invertebrate species based on things like hazard, exposure, resilience, status, value, and testability. I won't go into the details here, but there are nice examples where they've worked through these calculations for past biological control introductions that could be interesting for folks developing gene drives. It's actually interesting, this Pronti system was adapted from a scheme used to look at potential non-target impacts of transgenic plants. So this was biological control learning a bit from the transgenic organism development research community. Okay, so what types of post-release monitoring on non-target species are actually done in biological control? Because I tried to sort of break these down into categories. By far the most common type of post-release monitoring study in biological control are basically surveys of whether or not biological control agents attack non-target species in the field after their releases. But these studies of whether or not they attack them, whether there is an association or an ecological effect, don't necessarily tell us directly any information about whether that's having any long-term population level impact on non-target species. This is a theme I'll come back to again. The measuring sort of the population level impact of biological control releases is fairly challenging, so these studies are a lot more rare. I think what people envision when they think of post-release monitoring often is really long-term, sort of decade or more studies where continuous monitoring of non-target populations or communities is done replicated over space and time before and after releases. These studies in biological control have actually been very rare or basically non-existent. And we'll talk about why in a couple of slides. But just to highlight that actually, even though biological control has been around for a long time, as of 2000, the last time that there was sort of an accounting of this, there was actually only post-release monitoring data on non-target effects for less than 2% of over 5,000 biological control releases. So this reflects sort of, this history of biological control that I talked about, that it's only relatively recently that we've started doing this routinely. And so because it's only been in the last 15 years, there are just not a lot of long-term data sets available. So for these first two types of studies on this slide, I just want to give one case study each. I wish I had time to give more case studies because there's lots of interesting ones, but you can always reach out to me if you're interested in more references. But this case that I'll talk about comes from Europe, where this wasp, Pterimus sinensis, was released in Italy starting in 2005 against the Asian chestnut gall wasp, which is was a pest of chestnuts. And this is the target species for this biocontrol program. The non-target species were many other wasp species that make galls on oak and rose leaves in Europe. These are the non-targets. And what this study demonstrated was that after successfully suppressing the target host, the biological control agent was started attacking at least 15 different non-target species in the field. So this is something that we do not want to see after biological control releases. However, the attack rate was all at very low rates. And this is something that can often happen in environmental monitoring or post-release monitoring, is that we see the non-target attack, we see some sort of effect, but it's small, right? I just want to point out that doing this type of study was a massive effort. It was only for 3 years, but it involved collections from all over Italy, of a bunch of different types of host species that were poorly known before the study. But still, these sort of low rates of attack that were observed don't really tell us conclusively anything about whether this biological control agent was reducing the actual long-term abundance of these non-target species. So what type of data does it take to actually infer what we call population level impact? I'm going to give an example here from weed biological control, where this is actually a Canadian example, where this weevil, Mogilones crucifer, was released in Canada starting in 1999 to control a rangeland weed that was poisoning cattle, the hound's tongue, which is an invasive species, and the non-target species belonged to the genus Hekelia, which are these native plants. Especially in the US, south of the border, there was concern about an endangered Heckelia that could be at risk from this weevil. So this motivated a study to look at the impact of this weevil on these non-target plants. And they actually wanted to focus on, is it actually going to reduce the populations, the abundance of these plants? And a really nice series of studies demonstrated that the biological control agent did sometimes attack the non-target species in the field, so like observed in the previous study, but that this attack was transient. It only happened for a short amount of time during the development of the biological control program. It was spatially restricted. It only really occurred when the non-target species was in close proximity to populations of the target species and they spilled over. And when they actually worked through the sort of population ecology math of it, it did not translate into significant population level impacts. But just doing this study on this one sort of indicator species was a massive effort. It was an entire PhD thesis, actually, just to do it at 3 sites with some additional monitoring at 10 sites. Matrix population monitoring, life table experiments. Life table experiments are really the gold standard in biological control, borrowed from conservation ecology to determine whether there's actually population level impact. Mark recapture studies of host preference. But what I basically want to highlight with this case study is the massive effort it takes just to demonstrate population level impact of an ecological intervention like biological control or gene drives on a single indicator non-target species. Something to think about. What about indirect ecological effects? Because I'm limited on time, I'll go through this very quickly, but basically these are still extremely challenging to predict. We have these potential direct non-target impacts, but of course these non-target species are embedded within really complex food webs. They might be attacked by other things, and this might mediate indirect effects between the non-native biocontrol agent and other species in these food webs. Basically, the path that biological control research has taken, at least so far, is to mostly focus on minimizing these direct effects and only introducing very, very specific agents, with the idea that if the agent's very specific and there aren't many direct effects, that there also won't be many of these indirect sort of knock-on effects of the direct effects. But something it's important to point out is that this assumption is not always true, and this is something to think about. So this is an example from Australia, where a highly specialist seed-feeding fly was introduced to control this B2 weed, but it was ineffective. It did not suppress the populations of the B2 weed, and so it itself got extremely abundant. Insect predators and parasitoids in Australia then started feeding on these highly abundant species, highly abundant populations of the fly. That increased their abundance, and it was shown that they spilled over to suppress populations of native seed-feeding flies. So even though this biocontrol agent didn't attack these native seed-feeding flies, it indirectly acted to reduce their populations. And what this taught biological control researchers is you should not introduce an ineffective agent that's going to get to really high densities. And one of the things that one should worry about when doing these types of interventions is whether they're going to cause some species in the food web to get to really high abundance. And yeah, in this case, it was apparent competition. So again, this was a very nice study. There's a couple other ones like it demonstrating these indirect effects of biological control. But in terms of thinking about what it means of a post-release monitoring study, this was not a long-term study. It was done over a total of 3 months in a single year. So there's sort of a bit of a trade-off between the sort of level of insight you can gain into population level effects and how long you can do a given study. Okay, I'm on to my last 2 slides. First, I want to talk about some practical challenges for post-release monitoring that have sort of come up over the course of the presentation, but that I'm going to summarize now. So post-release monitoring, when it is done, is carried out by research labs with limited resources that are usually balancing multiple biological control projects. There's so many new invasive weeds and insects every decade that labs are usually balancing multiple projects with only just so many resources to devote to each. So this is one of the reasons why there isn't more post-release monitoring. And post-release monitoring efforts and resources are inevitably divided between measuring impacts on the target pest and the impacts on the non-target species. So this is something that I imagine will come up in gene drives as well, is how do you sort of balance the need to monitor the effects you want to see and the effects that you don't that you hope are not occurring. A huge problem for biological control is that our native non-target species that we're interested in can be hyperdiverse, poorly known, poorly taxonomically understood, difficult to find, and all these things make them very difficult to conduct long-term population studies on. Something we run into all the time is what we call the rare non-target problem. You're often interested in, from a conservation perspective, rare species, but because they're rare, they're extremely difficult to study. It's difficult to obtain sustained funding to do post-release monitoring. By the time you get to post-release monitoring, you're often 10 years into a research program, and it's harder to convince funders of science to fund something that you've been working on for 10 years than something that's brand new. And because there's so many new invasive species problems all the time, they tend to receive most of the attention and funding. And then sort of a scientific thing is that many biological control practitioners are organismal biologists, usually entomologists. They're not often trained community or population ecologists, and the science of biological control has actually advanced the most due to studies by critics of biological control who are invasive species ecologists or population ecologists. So something something the gene drive community could consider is recruiting its critics and really making sure that population ecologists are on board. And I know there's already a history of that in the development of gene drive technologies too. Some lessons for post-release monitoring based on biological controls experience. I think one of the main sort of messages I want to get across today is this distinction between seeing a potential effect and knowing that it has a long-term population level impact on non-target organisms. In biocontrol, we often look for the signatures of the effect first and then conduct detailed, for example, life table studies to see whether the size of that effect matters. The best insights, at least so far, have come from testing specific ecological hypotheses or mechanisms about non-target effects using tractable indicator taxa. So far, at least, the use of sort of generalized community-wide monitoring data where we sort of measure everything and then look for impacts after releases, it has not taken hold yet as a viable monitoring method. Even with the best of intentions, committing to conducting these complex and rigorous long-term monitoring studies of non-target effects may not be reasonable without assignment of responsibility. For example, if you don't have to conduct post-release monitoring, it may not get conducted. And then a huge issue is a lack of guaranteed dedicated funding support to be able to commit to doing non-target monitoring for years after releases. And I also just want to highlight that it's not as if in biological control, even with a more than 100-year history, this is something that researchers have sorted out. The need to conduct more and better post-release monitoring is still a major issue in biological control research, and it's still routinely called for by both advocates and critics. So that's one of the reasons why I'm interested to be part of the group today and next week, because I think there's things that I can bring back to biological control research from learning from you all as well. I want to thank everybody at FNIH and Gene Convene, David, Stephanie, the whole team for inviting me to speak today. I want to thank the researchers, my colleagues around the world who volunteered information about post-release monitoring, and please feel free to reach out to me with any questions. Thank you very much.
Thanks a lot, Paul. Really illuminating talk, and I think highly relevant to the task at hand today. So let me thank Paul, but also let me encourage the audience to chat in your questions at any time for Paul, and really encourage you to do that so we can sort of encourage Paul to probe some of these things a little bit more at the end of the Q&A session. Renee, I'd like to invite you to Renee Bellinger. I'd like to as our next speaker, and I'd like to invite her to take the floor. Renee. Go ahead and put it on presentation mode, and yes, you are, and I can see it just fine, Renee. Thanks for being here. The floor is yours.
Thank you for the invitation, and thank you, David, for organizing this webinar series. It's a real pleasure to be here, and I appreciate everyone's attention during this presentation. So my name is Renee Bellinger, and I'm a research geneticist at the United States Geological Survey Pacific Island Ecosystems Research Center located on the island of Hawaii. And today I'm going to describe a monitoring and biosecurity program that we've designed as part of a genetic control technique using Wolbachia, cytoplasmic incompatibility, and applying this technique as a conservation measure to suppress invasive mosquitoes that carry avian malaria, which is having devastating consequences on our forest bird populations. I'm going to start with the big picture view to frame the uniqueness of the Hawaiian archipelago. All right, so Hawaii is located in the middle of the Pacific. It's one of the most remote islands in the world. It formed de novo in the middle of the ocean through volcanic processes. And because of the distance of the islands from continental areas, there were few founding events by plants and animals. And the plants and animals that did arrive to the islands encountered a novel landscape. And that released them from the constraints that they had from where they came and allowed for ecological opportunity to diversify into forms. And that led to a number of unique adaptive radiations that are characteristic of these islands. And so there is where we're located at. And because of this distance from the mainland, we actually do not have any native mosquitoes to Hawaii. And it's one of the few oceanic land masses that doesn't have any form or any type of mosquito that is native. However, 6 species of biting mosquitoes have invaded over the course of the past 200 years. It was first recorded as a singing fly around 1826 with the accidental introduction of Culex quinquefasciatus, followed by Aedes aegypti in 1892, Aedes vexans nocturnus in the 1960s, Wyomia michelii in the 80s, Aedes albopictus in 1986, and Aedes japonicus in 2003. Today, I will be focusing on the ecological impacts of Culex quinquefasciatus, that southern house mosquito. So after having arrived in 1826, this tropical mosquito rapidly spread throughout the islands in residential and agricultural areas, and ultimately into the lowland native forest. Declines in native bird numbers were observed at the turn of the century, along with the first records of bird pox lesions in native birds in the early 1900s. And you can see the impact of the avian pox on the bird's foot there in the middle. So continued population declines and species extinctions followed. The first detection of the avian malaria parasite was reported in 1940, but likely it had been around for decades earlier. The Hawaiian forest birds in the 1960s and 1970s were starting to disappear from the lowland forest and were becoming restricted to the high elevation forest where it was assumed that the southern house mosquito could not become established. However, with current climate warming trends, the reach of these mosquitoes and the diseases that they carry is increasingly extending into higher elevation areas. Historically, there were greater than 50 species of honey creepers on the main Hawaiian islands, but today only 21 species remain, and more than half of those are federally listed as threatened or endangered, and of these also the IUCN lists 19 as vulnerable or worse. 4 single endemic bird species are at high risk for extinction. In 2021, the estimated time to extinction for 2 species was believed to be within a year or 2, and for the other 2 species within the next decade. At this point, the akikiki, shown to the left, is likely already extinct in the wild. By last count, I heard 1 individual was known to be in the forest, and the rest of the species had already been brought in for a captive rearing to try and not lose the species altogether. So concern for the continued declines in the extinction of Hawaiian forest birds led to an ambitious task, which was to rapidly develop and implement a population suppression strategy for Culex quinquefasciatus as a measure to disrupt the avian disease cycle. Our group is called the Birds Not Mosquitoes Coalition, and it's comprised of more than 15 federal and state agencies, NGOs, and private enterprises. The technique that was selected for mosquito suppression was to use incompatible insect technique, which leverages the impacts of Wolbachia, a naturally occurring bacteria that lives in cells and some nematodes, and its impacts on the reproduction of its host. Wolbachia in some strains can cause cytoplasmic incompatibility, which simply means that if you have two incompatible strains of Wolbachia in a male and a female, the eggs don't hatch. They basically die through embryonic death. And CI is what's being leveraged for the incompatible insect technique, resulting in that embryonic lethality. And of note here, Culex princifasciatus naturally carries Wolbachia. So in this program, it was necessary to generate a bidirectional cross, meaning that the males couldn't mate with females and vice versa. And then the idea is that through repeat inundative releases on the landscape, the population of mosquitoes will eventually decrease and then crash, and that should disrupt the avian malaria disease cycle. This coalition started its work in around 2017. By 2021, we had began the import permit process. We had gone through the paperwork. We did a lot of public scoping, public comment periods, and then in it was late 2023, we started to conduct field tests and then began our implementation in a select area on the island of Maui. And now in 2024, we are releasing mosquitoes on the landscape. And there are plans to expand this program to the island of Kauai. And then there's import permits for the remainder of the state as well, and for other species for human health purposes. So genetic biocontrol encompasses both Wolbachia and gene drive. And I just wanted to point out some of the similarities between the two. Wolbachia is naturally occurring, so that's dissimilar, whereas gene drive is an editing of the genome as a living modified organism. Both techniques can skew the sex ratios of the host. It can alter reproductive fitness and alter phenotypic traits. In the case of Wolbachia, it's a host-bacteria interaction, which in some cases is beneficial and in other cases is more parasitic. And then with gene drive, of course, you can do a targeted alteration to phenotypic traits. You can impact disease transmission using either technique. And the Wolbachia IAT program has a fairly straightforward regulatory pathway that is well defined. We have the EPA, so national EPA, Hawaii EPA, state microbial permits, import permits, and the regulatory pathway for gene drive is much more complex. And because Culex is not native to Hawaii, and because the Wolbachia albopictus B, which is the strain that is being utilized for this program, is already on the landscape in Hawaii, this program is not believed to have major impacts on the species that already occur. Also, the mosquitoes are not considered to be a major component of any diet of any organism. And because they were not here before, should they become eradicated, that would not impact or perturb a system where they naturally occur. Now, of course, in the case of gene drive, depending on the application, the native status of a mosquito would be case dependent. So for building this Wolbachia incompatible insect technique program, of course, we needed to get a Wolbachia that was bidirectionally incompatible. This work was conducted through partnership with Verily. And the little green dots there is the Wolbachia inside of an insect cell. And again, both the males and the females, when they mate with each other, the eggs will die. We have to go through the mass rearing and then Verily conduct sex sorting and the male selection using like a sieve and then their AI technology and a classifier. And they also developed a distribution and packing system. So these are biodegradable little pods that are distributed with the mosquitoes on the landscape. Of course, we had to select sites. These are around 3,000 acres total, so somewhat small scale relative to the entire landscape, but these are high impact areas with the Hawaiian forest birds. We have a distribution where you see these little alternating teal and brown dots over the landscape. Those are our pod distribution sites. And for deployment logistics, these are very rugged, remote areas. So that area that is shown for the site is actually up on a shelf. So you can't just drive to it or walk to it. And for that reason, we use helicopters for the application at this point. And those two yellow arrows indicate like a little gun, and then the pod that is popping out of the helicopter. And then we're going to refine the application through the use of autonomous vehicles. And so naturally, we want to be able to establish whether or not our IAT efforts are successful. We want to monitor for biosecurity, and then we need to be able to come up with metrics for showing that the program worked to reduce the disease of the avian malaria in the birds. And so we've conducted a number of types of adult mosquito trapping studies. So in one of our studies, we evaluated the efficacy of three trap types for monitoring adult mosquito abundance. That work was conducted by Catherine McClure and Dennis Lapointe. We developed a field protocol to estimate egg raft viability as an alternative metric to IAT efficacy when mosquito captures do not provide enough power to detect change. And then also these oviposition cups are necessary for the Section 18 authorization for the monitoring, and the eggs are basically hatched out and then sent to Verily to check for any of the Wolbachia albopictus B, which is the transinfected strain in Culex. This work was conducted in the Kipahulu Valley, the site that I showed in the last slide. And then we collected water samples for the development of eDNA protocols, and we are continuing to refine those as we go. We also tested different types of lures. So a BG Pro Trap baited with carbon dioxide, a BG Sentinel trap. We used wing beat frequency lures and other types of lures as well. So these oviposition cups, I just wanted to highlight those there because that's a part of our required monitoring. And then for eDNA sampling, our goal was to develop an eDNA sampling technique that was easily deployable in the field and relatively inexpensive. And so we developed our species-specific primer and probe. We utilized the Culex COI gene. We conducted in silico analyses and then in vitro and in situ testing. When developing an eDNA tool, you have to test the assay efficiency, limits of detection, and limits of quantification to ensure that the tool is sensitive and specific to the organism that you are monitoring for. We did a lot of testing in the lab just by rearing up mosquitoes. And then we field validated this by comparing the mosquito eDNA results to the stream dip surveys and the adult trapping data. And we have a flow chart where we have a decision matrix in terms of how to quantify whether or not we decide that a sample is positive or negative for an eDNA replicate. We're running 6 replicates. Sometimes you only get 1 or 2 samples that will actually amplify. And so we go through a process to determine what is it that the eDNA is so low concentration, or do we need to tweak the reaction, or are there inhibitors? And so inhibitors can come from the environment because we have a number of different types of pools that we're sampling. Some have mud, some have leaves, some have a lot of organic material. And so shown here is some of our mosquito monitoring in Kipahulu Valley. You can see the mosquito trap, and that red circle line is one of the eDNA sample pools. And then to the right is like a little dip cup. So the field crews went out, they dipped the cup in the water, they looked for mosquito larvae, they reared up the mosquito larvae, and they took the eDNA samples from there. And we tested 3 different filter types to determine the efficacy, but I don't have results for that just yet. We did detect Culex in the Kipahulu Valley, and then those were mostly at the low elevation sites, and at the higher elevation sites, there were 80s albopictus to be specific. So in total, there were 12,000 dips in this surveyed habitat. It's actually quite difficult to figure out where the mosquitoes are breeding. And so we're hoping the eDNA will lend some insights into the seasonality, like when we're missing the mosquitoes versus the mosquitoes actually aren't there. And so to date, we've collected around 180 eDNA samples. Again, we're looking at these different filter types, and then we're testing a variety of different sampling techniques, trying to find the one that is simplest to implement, that has the biggest bang for the buck. And those yellow circles there is preliminary data showing areas where we have positive eDNA samples within the basin, which differs from what we saw in the dip cup results. And just again, reiterating, when you're trying to detect mosquitoes in diverse aquatic habitat, there's actually a lot of challenges that you have to overcome in terms of accounting for the inhibitors that might be present in the water itself. So moving on to the next type of tools for biosecurity, when the adult mosquito traps were set, we collected all of the adult specimens and subjected those to genotyping and to also Wolbachia strain typing. And then in terms of programmatic efficacy, this is work that is in progress, and that would be to model the mosquito abundance and avian malaria disease response. And ideally, we want to have an epidemiological outcome that we can quantify through a reduction in disease. But it's challenging because the birds have a lot of reservoirs for the avian malaria within the system. And then the birds, the Hawaiian honey creepers, actually live longer than other passerines. They're like up to like 10 years plus. So they're going to be potentially harboring avian malaria, and we don't know, or cannot discriminate whether or not a bird was infected or it's just like a legacy infection. And then we can also monitor the mosquito's response using traps and genomics data. So calculating relative abundance and then looking for signatures of differences in the genomes of the mosquitoes. And so with regards to QLEX genomics as a genetic baseline, we sampled 37 populations for around 3,000 specimens across the Hawaiian archipelago, and we genotyped 950 of those using a DD-RAD or a reduced representation protocol. And we found that there's pretty moderate structure between islands, but within islands, even despite sampling from low and high elevation sites, there just wasn't a lot of local structure. And that might be because the mosquitoes have rapidly established on the landscape and they haven't yet differentiated in their kind of pockets where they're residing. Or it may be that they're moving around a lot because QLEX in particular is a able-bodied flyer. And then we're continuing our modeling and analyses. We did look at FST, so this is a measure of allelic differentiation across the genome, just looking for divergence and for outliers that might be indicators of either differences between populations or regions of the genome that are under selection. And so these are just results for two different islands, one on two pairwise comparisons, two on Kauai and two on Maui. And you can see that on Kauai, this, I don't know if you can see my cursor, but the FST values are very low. And then it's moderate in these other pairwise comparisons. And then there's one population that stood out and showed some diversification. But in general, they just weren't very telling. So unfortunately, that means that we are not able to have strong confidence in utilizing the genetic information to identify, say, population sources when we're doing our treatments. We're continuing these analyses, though, looking at comparisons across the genome and again, looking for genetic signatures of adaptation. These ones are pairwise comparisons within islands. And then these are between islands, the ones I just popped in. And so the upper four on the right-hand side does show a greater signature of divergence. So we may be getting close to being able to identify regions of the genome in some cases that are under selection. And then another consideration when studying mosquitoes, in particular QLEX, is these species have 3 chromosomes and they don't have an XY chromosome. They have an M locus or an M factor. So that's the determining factor for the sex. And so we looked at the proportion of shared alleles to evaluate whether or not there's differences between chromosomes in the similarity of individuals across the genome. And so to the left, you can see that the colors are slightly more yellow, which means that they're slightly more shared. These alleles or the genes themselves are more shared on chromosome 1 for all of these individuals that were genotyped. And this is a total of, I think, 11 different populations, and each little box is an individual compared to another individual as a matrix. And chromosome 2 and chromosome 3 have less shared alleles. So when designing, for example, say we were to go down a synthetic biology route or a gene drive route, chromosome 1 is going to be a more likely target, both because of that sex determining factor, but also because it's more fixed, if that is considered an attribute for the technique. And so for the QLEX, we took the same data set and looked at Wolbachia and asked the question whether or not we could compare the Wolbachia genetic signature to the QLEX genetic signature. And then also we're using the QLEX genomics to determine whether or not we can detect like population reduction in sizes. And that the population bottleneck effect should be reflected in the next set of samples that we sequence, which will be whole genome sequencing. Okay, so next I want to talk about Wolbachia diversity and tools to monitor for CI resistance and test for Wolbachia introgression. And so, as I mentioned, we have moderate population structure of mosquitoes across the 3 islands. So we use that same DD-RAD data to look at Wolbachia. And in fact, there's a lot of diversity with Wolbachia, but they are not structured by island. We do have evidence that there's 3 different lineages of Wolbachia in Hawaii. And so one of the questions that we have is whether or not there might be some resistance to the CI that was developed for this program. And that's kind of akin to what you would want to test for a gene drive system as well. So in the case of complete cytoplasmic incompatibility, you're not going to have any eggs hatch. In the case of incomplete cytoplasmic incompatibility, you might get a certain proportion of eggs that hatch. And if that were to happen and those individuals were to have that, if they live, which they might, then they would breed within the population and the genetic background of the mosquito that was being released would introgress into the genome of the wild type mosquito. The mosquitoes that are being released for this program originated from Oahu, and based on our other genetic structure information, we anticipate, but do not yet have evidence, that Oahu population genetically differs from the populations of QLEX on Maui and on Hawaii. And then in the case of female release, if that were to happen, then that Wolbachia WLB could potentially get released on the landscape, and that would reduce the efficacy of the IAT program and could in fact render it ineffective altogether. So that would be like a resistance. I have this crossed out because it is there is cytoplasmic incompatibility. And so the female would have to mate with one of the released males in order to even establish on the landscape. And so we are monitoring for this female release. Verily is looking at the egg grafts using a marker that they developed in a PCR, like a qPCR. And then I'm more focused on the incomplete CI and looking for signatures or changes in the Wolbachia composition and the mosquito genome. And so as a part of the Wolbachia biosecurity monitoring gene panel, we've taken the genes that are known to have involvement in cytoplasmic incompatibility, in particular SID and SID-A, SID-B, which should be CIF, following nomenclature that is accepted, that is recommended. We developed a gene panel using a technique called GT-seq, which is genotyping by thousands, so that we can rapidly screen mosquitoes from all over the archipelago. And then we're expanding these collections on a global scale so that we can characterize the Wolbachia diversity within QLEX, and then also see if it works for some of the other closely related taxa. And this can be applied to assess the IAT integrity as part of biosecurity monitoring, and also to identify outlier mosquito populations that might be prone to an IAT failure. And our next-gen sequencing is in progress. And here, this is not to read, but we have all of these different genes. They're actually quite long, so we had to do overlapping primer pairs in order to implement this monitoring technique. And so the same type of approach could be utilized in a gene drive system where you want to rapidly sequence large volumes of mosquitoes to test for introgression of a particular gene. And then in our case, we have a MySeq, so we're conducting kind of mid to long read, well, it's mid read, 300 base pairs, so 600 amplicon length sequences, and then we'll use those to conduct phylogenetic analysis of the Wolbachia. So in terms of lessons learned from the field, both Wolbachia and IIT, and also gene drive, requires an understanding of host ecology. We have a lot of tools to monitor IIT and gene drive using genetics. For example, we can measure the introgression of genes. We can monitor for resistance. We can use these same tools to monitor or detect weaknesses in a mosquito control program. But the tools to model the programmatic efficacy really are custom tailored. And with that, I want to thank my colleagues, Catherine, Carter, Dennis, Eben, and Karina, and acknowledge that this work is a partnership with many groups in conservationists. And I'm going to stop here with my slide as with my contact information, should you have any additional questions as a follow-up.
Great, thanks a lot. Thanks a lot, Renee. Really interesting and actually very exciting too. And so again, I encourage the audience to chat in some questions for Renee. We'll get to them at the end of our final presentation. But while her presentation is fresh in your mind, please challenge her with some questions so we can bring out some more details about what she's doing and some of the challenges they're faced there. So our next speaker is Jafet Osuna from the University of Glasgow's School of Mathematics and Statistics. So Jafet, I am encouraging you now to take the floor and to go ahead and share your presentation.
Hi, David. Thank you.
Good. I can see your presentation.
Okay, that's great.
It looks good, and I can hear you. And the floor is yours. Thanks for being here.
All right. Okay, thank you so much, David. All right. Hello, everyone. Thanks a lot for joining today. Let's just so well, as David just said, I'm Jafet Belmont. I'm a lecturer at the University of Glasgow at School of Maths and Stats. And today I'm going to talk about some of the research I've done in the past 2 years with some other collaborators from Glasgow and Norway as well. And specifically, I'm going to talk about some of the challenges that we have identified while addressing different sources of bias in biodiversity monitoring schemes. So, right. So just as a short overview, well, we know that biodiversity monitoring programs are essential to describe, map, and predict species distributions. These are crucial to understand where species are in space and time. But as you heard from previous speakers, collecting data can be very costly, especially for large-scale studies. So having planned surveys require a lot of effort, sampling effort, and resources that we don't always have or are available to us. In that sense, different large-scale monitoring schemes, such as data from citizen science, offer the cost-effective solution to investigate species distributions across broad spatial and temporal scales. However, analyzing such data is challenging, partially because there are a lot of sources of uncertainty that observe the process of how data is collected. So here are just listed some of the main sources of bias that are typically observed in such class of surveys. So we have, for instance, preferential sampling, which refers to the fact that people go more often to places or locations where they know that species are going to be found or where the abundance of those species is high. So the sampling locations are biased towards those survey locations. We can also have misidentification error, having incorrect classification of a species, specific species. We have also detectability. We can often miss or not detect species even though it is present in a given location. And we also have varying recording effort and recording skills among the participants in these surveys, and uneven sampling effort, both over space and time. So all these sources of error affect how we observe the data, and that basically affects the ecological process of interest we want to model. So we only have a partial picture and complete picture of where species are and why species are there. This ecological process of interest is also affected by different geographical and environmental conditions, which themselves can also impact on the observational process. So if you go on a very rainy day, for instance, to the field, you might not find certain species, but that doesn't mean that the species are not there. But at the same, rain conditions can shape the species distribution. So we need to consider or take all these into account and factorize it in order to provide a complete picture of how all of these features can impact different health, social, and economic futures. So there are a lot of advantages from these large-scale monitoring programs. So for instance, they offer an extensive taxonomic and spatial temporal coverage that very often plant surveys cannot have. They can offer a monitoring over longer time periods and across large spatial scales as well. And specifically for those species with eye-catching traits, those species can be easily identifiable by participants with a relatively small amount of training. So that's like some of the advantages that we have when dealing with such data. Now, of course, there are some drawbacks from such large-scale studies. So, for example, we can, and it's very common, underreporting of certain species, especially if they're rare or hard to detect. Those species are going to be difficult to spot. And also we have varying recording effort and skills among the participants in such surveys. So we need to take into the account those sources of errors in order to provide with an accurate descriptions of where species are and how to predict where species will be in the future given the environmental information that we have. So the first part of this talk, I'm going to discuss on the reporting of some of the species. Particularly, I'm going to talk about the problem with imperfect detection. That is a problem of a species not being detected, even though they are in a location where we go there and survey that space. So I'll illustrate this problem of detection in the following picture. So try to have a look at the picture and find out how many different species there are. So as you can see, detection is not perfect. In fact, in this picture, we have 4 different species. There are some bird species here poaching a tree, a leapfrog, some caterpillar, or even a gazelle. So detection is rarely perfect or constant because it varies with species rarity or with observation, with the observer skills, or even with environmental conditions such as you can say, the vegetation coverage in these particular cases. So it's important to identify this source of bias when we are doing monitoring. There are different issues if we ignore this important source of bias. For example, we can underestimate the true species distributions, or we can bias the estimated relationship between the species responses and the environmental information that we have in hand. And if we produce models that predict where species are going to be, we might end up reflecting detection rather than the true occurrence of the species. So from different methods that have been developed to address imperfect detection, occupancy models have proven to be very efficient to understand and describe species distributions while correcting for imperfect detection. The basic idea of these models that we've been working on is that data contains information about 2 different processes. So on the one hand, we have the ecological process we are interested about whether a species is occupying or not a given location. And that occupancy state depends on the occupancy probability, which can be modeled in terms of some environmental information we have available. So that's the true process where the species are in space, and that's one of the model's subcomponents. Then what happens is that people go and survey those locations. And then we have that condition on this occupancy, on this ecological process, we have an observational process that describes how data is collected. So across different sampling locations, so these models relied on multiple visits to be carried on during a site, and it's conditioned on the true occupancy state. So we are not going to be able to find a species if it's not there. But if the species is there, we might not detect it across repeated locations. We might detect it at least once, like in this gray square area, that the species is truly there, and we detect it on the first visit, but we didn't detect it on the second one. Or we might not detect it at all, even though it's present, which is a false negative outcome, like in this square area where this species is actually present, but in the two occasions that we went there, we never see it. So that is the data that we collect, and this is the picture that we want to reconstruct based on the observed data. And of course, these directly observed data are modeled based on the repeated visits we have and some environmental information or information that can explain this detection, which basically allows us to estimate those detection probabilities. So basically what we are doing here is we're trying to infer or we're making inference on a set of discrete sites, let's say AM in this particular case. And we are relying our inference based on the detection histories of each site of whether a species has been observed or not across repeated sampling locations. And therefore, the way we estimate those parameters is through a likelihood that looks something like this, but has two different components. On the one hand, we have the likelihood of those sites where we have seen the species at least once. So we know that the species is present with a probability phi, but it's either detected with a probability p or not detected at all. And then we have the second part of the likelihood for those locations that we've never seen the species. So there are only two options. Either the species was present here with a probability phi are not detected, or the species was truly absent with a probability 1 minus phi. So what we in our group have been doing is to incorporate a random effects for the probability of occupancy, because that's the one we are interested into. So we want to model spatial and temporal components as well as non-linear covariate effects to make the model more to capture things that we usually see in reality in real case studies. So we are using an inferential approach called INLA, which is for integrated nested Laplace approximation for a class of models named latent Gaussian models. These models have three main components. On the one component, we have what's called the random field, which you can think of this as all the different random effects that you have in your models. Then we have that depends on certain hyperparameters there. And then we have the model likelihood that has a linear predictor linked to this random field over here. So INLA, it's a fast and accurate Bayesian inference approach that allows us to estimate such latent Gaussian models. So that's basically, on a nutshell, the tool we are using to estimate those parameters. And after some formulation, we can show that these occupancy models can be formulated as a latent Gaussian model, and therefore we can use INLA to find or estimate those model parameters. And the reason why we are using INLA is because it's fast and allows us to incorporate large spatial effects that other kind of packages are kind of limited in terms of computational efficiency. So if you have a large collection of sites or a large number of species, the usually available tools that you have to model such or to fit such models are not computationally efficient, and you can be limited by that. So in this case, we're using this, and we developed this framework in this inferential approach to speed up things in a computationally efficient manner while providing a lot of flexibility in the things that we can do. So for instance, we can incorporate random effects in the linear predictor, so we can incorporate space and time dependency. And it's relatively easy. We have there's R in the software which you can use to implement such to give a fast and reliable algorithm. And just recently we have expanded this to incorporate Harvey level covariates as well. So if you go on a rainy day or on a very windy day and you register that information in, you can also incorporate that into the model itself to correct for the fact that your species might not be detected on a very bad weather, on a day that's on a very bad weather. So you can incorporate that information in the model. And now I'm going to present just just an example of the applications in which we quantified some spatially varying trends for these great catbird species from the North American breeding survey, bird survey. So basically, we took 2,000 locations that were sampled between 2000 and 2019 for these birds, where this species was being recorded. The records for this, the presence of this species have been recorded, and across different bird conservation regions as well in the US. So we have identified those bird conservation regions and found 2,000 sites where the species have been observed. So these are just the yearly detections per location in that survey. The number of counts were how many times the species was seen. And so we propose this model where we model the spatial temporal presence absence, true presence absence of the species. And we propose 3 different models here. And the first one, it just assumes that there's a fixed time trend. The trend is constant over space. The second model expands on this and allows us to fit a spatial varying trend on a continuous space framework, plus some spatially structured random effects. And the third one, it's another model that allows us to fit a regional varying trend across each of those bird conservation regions. And then for the observational model, we allow the models, the detection probabilities vary according to the date in which that survey location was visited. So this is the typically output you get from fitting such models. You have information about the occupancy, the true ecological process you're interested in this particular case. You have the baseline occupancy. In this case, it's just the fixed trend that was estimated. Then you have a detection parameters. You have some model comparison metrics and how much time it took to run such models. So for instance, we've seen that even the most complex models, which is this one with a spatial varying trend, took approximately 2 minutes to run, whereas if you were looking to some other software that it's available, it would probably take you weeks to run. So these are some of the results that can be used to communicate the outcome of such models. So the first one, it's just the predicted temporal trend on a constant basis. So this is the model that assumes there's a constant trend. And if you just fit this model, you will be basically estimating a negative trend in the population. However, when we look at the second model, we've seen that there are actually locations where the trend is actually negative, but there are also locations where there's a positive trend. But for most of the country, that trend it's not significant. And the last models, if you are interested in particular areas or geographical areas, you can summarize that information on a regional level. And so this regional model we developed identifies those regions where the trend is actually significantly negative, and those regions where there are actually no change at all. So those highlight some of the outputs you can get from fitting such models. We have also the predicted occupancy probabilities in 2020 based on the data we got from each of these 3 different models. So in summary, this inferential approach that we developed, it's powerful and flexible, allows you to incorporate complex random effects for the occupancy probabilities in this case. It's fast and reliable, and great for analyzing a standardized or a protocolized surveys. It allows you to correct for imperfect detection. The next question is obviously, okay, what happens if I have a partial or complete lack of design in my protocolized sampling schemes? And what happens in citizen science data where we know that sampling effort is not even? Well, that's the second part of this talk about some of the other challenges that we have addressed. And particularly in large-scale monitoring schemes, sampling effort is not constant. We know that participants involved, for instance, in citizen science, visit certain locations more than others, mainly because accessibility bias. But we need to make a distinction between the reasons why sampling is biased in space or time. Very often it's the choice of locations is biased towards external sources to the outcome of interest. So they are independent on species distributions, for instance, or the species abundance. You could go look. People go to locations where they do their activities. So that's basically for the most cases independent from the species distributions. And there's another bias which it's usually referred to as preferential sampling, where people choose locations where they know or where they expect to find the species, or where they know that the species abundance is high. So there are two different sources of bias. In this talk, I'm going to discuss the first one, which is a more common source of bias. But the second one, it's also an important source of bias that needs to be considered as well. So how can we go about this sampling bias? Also, we call it a accessibility bias, kind of make a distinction from the preferential sampling one. And well, it's been proposed that if you use collective records from multiple species, this can be efficient way of reducing the effects of sampling biases. So citing this paper, the idea is that the surveyors or participants in these schemes will display the same bias when sampling similar species, and therefore a large data set with multiple species will contain information on the general sampling effort for a given taxonomic group. So this is just an example from a birds conservation monitoring scheme, where the red points illustrate locations where a focal species that the species we're interested in has been observed, and the blue points are locations where any species other than that focal species have been observed. So we can get a full picture of the whole environmental space, and that's the idea, and we get an idea where people are going. And nowadays, with a large volumes of data being available for multiple taxonomic groups, based from different observations or different sources of information, we can get very large databases and very large data sets to get an idea of what the sampling effort in different schemes is. However, there's also another problem, and it's that each data source comes with their own biases. So the next question we have is, can we gain something by combining the information from multiple surveys? So that's what I'm going to address in the next case study. So for this one of our PhD students was working on the distribution of these specklewood species, butterfly species, found from and records obtained from this WCBS, the Wider Countryside Butterflies Survey Scheme, which basically targets types of habitats that are usually under survey compared to other sampling schemes. And then we also use information about the Butterflies New Millennium opportunistic survey. This is one, it's a complete opportunistic survey where just people go out wherever they want and report whatever they found. So we are getting the information from these two different sources. This is just an illustration of the data set we're using. So we're matching the records on a 1K grid reference level, commonly used in the UK or in Scotland. And we are basically using the same temporal support for these two data sets. So on this side, we have the information from the opportunistic survey. On this one, we have a bit more structured plant survey for the wider countryside one. The red points are the locations where these specklewood species have been found, and the blue points are the locations where any other species have been observed, basically where people have also been there recording other species. Now we proposed a point process model to analyze these data sets. So we propose a framework that can be used to correct for sampling bias. So the first idea is that we will create a joint model for the point pattern that describes the locations where any species have been observed. Basically, use this as a proxy of where people are going in the field. And so we model this as a point process where intensity function would basically measure the contribution on the expected number of individuals that are going to be found in an area. And so we model this through some environmental covariates and some spatial structure effects, because commonly all the covariates won't account for all the spatial variation that observed. So we need to incorporate this information to account for those unobserved environmental features. And then in this case, with this, we'll basically capturing those features that explain the sampling process. So everything that's not accounted by the credits you have here to explain where people are going are going to be captured by this omega term. So this is basically a proxy for where the sampling bias is going, essentially. And then we're modeling the records of the species of interest of this specklewood butterfly species at another point process whose intensity have similar or different set of covariates, but with different parameters to be estimated here. We have its own spatial effect that basically describes the underlying spatial structure for that species. And we have this shared omega term here, which is scaled by this phi parameter, which measures the contribution of this unexplained variation from the sampling process into the process of species of interest. So we are factorizing the variation of the sampling effort into the process of interest to account for this sampling bias. And then we have produced a final joint model for these two survey schemes. So each of the survey schemes have their own sampling bias process, I have described in these questions 3 and 4. And then on equations 5 and 6, we have the true process of interest for the species we're looking at. So we are producing a joint model. We have shared effects, these spatial structure effect and covariate effect. We assume that this is the true that we were looking for. And therefore, we are just simply correcting for the source of bias of each data set here. So we are factorizing the source of bias of each independent data set while estimating jointly the true parameters of our species of interest. So these are just some preliminary results we have for each individual model fitted. For the models fitted on each data set independently. So on the left-hand side, we have the New Millennium data set where we can see that there's a much higher intensity of the species in the northern of Scotland. And then we have the other survey scheme where we have a higher intensity near the regional park and the Inverness area. And finally, we have the results for the combined model that uses this information from the two survey schemes while weighting in the sampling process of each independent survey scheme. So we can see this is the kind of like the full picture, and we can see how the field of the explained variation of the sampling process influences the estimations of the species intensity through this sampling parameter, which is a greater than R0. So there are some biases there on the sampling process that affects the species intensity in different ways depending on the survey scheme. So just to conclude this section, we've noted that these point processes are a general tool that is flexible and can be used to combine data sources with their own source of bias. And we have extended this approach to account as well for preferential sampling. Now we want to exploit the fact that in most of these schemes, a lot of these grid cells are surveyed across multiple locations. So we can extend this framework and actually use this to correct for imperfect detection as well. That's something we're currently looking at. Things to do, of course, we need a way of validating the results and do some model comparison. So far for these point process models, we still rely a lot on on experts to tell us whether the results make sense for the species we're interested into. But we would like to produce some more quantitative metrics to allow us to do more a quantitative model comparison between the different methods or models we developed. Also, we like to develop a joint model weighting approach that will basically weight the contribution of each data set, because as you've seen in one of the pictures, the data from opportunistic surveys is vastly and has a great coverage, but this might outweigh the information you get from smaller sampling schemes, even though these smaller ones could be planned surveys where you have less amount of bias. So there should be a way in which you can tell, okay, I have less data here, but this one is actually more reliable than the other one, even though in the other one you have tons of data. I know this one is more precise, so we are working on working ways in which we can add this information into the joint modeling approach. And the other thing we noticed that as well is that some of these biases are scale dependent. So, for instance, we assume that this preferential sampling or the sampling bias occur all across space, but that might not be realistic. This might happen only in certain areas, for example. And so we are extending this approach to model these sampling bias parameters, to allow these sampling parameters to vary over space, to essentially say, okay, this doesn't have to be true for your study area. This might have happened locally in certain bits. So that's also something we are currently working on. And that's it for me. Thanks a lot for listening.
Great. Great. Thank you, Jafet. Really appreciate the presentation. Again, let me encourage the audience to go ahead and chat in questions as you think of them for Jafet or any of the other previous speakers as well. I'd like to now welcome our final speaker, Sarah Paul, who is at the National Ecological Observatory Network in Boulder, Colorado. So, Sarah, the floor is yours. I can see your presentation.
Great. Thanks so much.
Take it away, and I can hear you as well.
Awesome.
You're good.
Wonderful. Well, I'm really excited to be here. Thanks so much for the invitation, and thanks to everybody who is joining in. I'm really excited to introduce you to the National Ecological Observatory Network, for those of you who are unfamiliar with what we do. So this talk, I'll start out by talking about who we are and what we do and what kinds of things we sample. And then I thought it might be useful to discuss a little bit of the history around how we started and how we made some of the decisions about how to monitor what we look at and when to do that sampling. And then I'll finish up by talking about some lessons that we learned along the way. All right, so NEON. NEON is short for the National Ecological Observatory Network. We are a continental scale, long-term observatory. We're funded by the NSF and we're operated by Battelle. And what we do is we essentially are here to enable free and open data and samples on drivers and responses to environmental change. So one of the most unique and important aspects of NEON is that all of the data that we collect are collected under the same standardized framework. So that allows us to pool all of our data sets to make continental scale comparisons, as well as over long-term time periods. And we also work really hard to have interoperability of our data. So when we formed the observatory, we worked a lot with partners that are also collecting these long-term data sets to make sure that our data would be compatible with the things that they're collecting to enable these very large scale data sets for baseline analyses of ecological patterns. And so one of the big questions is, why is NEON important? And so I'll get to a little bit more of this when I talk about the history. But one of the most unique aspects of NEON is that we're a highly coordinated national system for monitoring. And so essentially what that means is that all of our protocols are standardized, and we work really hard to make sure that data are being collected in the same way across all different areas in the same way from year after year. And so this is really allowing us to make the data comparable so that we can combine them and get these continental scale analyses. And so NEON, this is basically just a map of where our sites are located. And you can see that it's continental scale. So we're talking about the continental US, as well as Alaska, Hawaii, and Puerto Rico. And we have 81 total field sites, and those are split between 47 terrestrial sites and 34 aquatic sites. And so you'll notice on this map, there's these blue outlines, and those are corresponding to ecoclimatic domains. And essentially what we ended up doing to decide where to locate our site so that we could get a monitoring facility that would really look at the entire United States in a really comprehensive way was we grouped into 20 different regions according to similarities in climate and ecological variables. So there's at least one core site and one to several gradient sites in each of these 20 domains. And I'll talk about this a little bit more later, but core sites are in wildland areas, so they're sort of the baseline comparison. And the gradient sites were selected to measure the same variables that are being measured at the core sites, but they represent a gradient in anything from agricultural practices to forest management types. And we collect data on over 180 different variables that are in the environment. And this ranges from temperature to soil properties to plants and animals and other organisms that we might see. And the intent is to be collecting data across these domains over 30 years. And so our operation start date began in 2019. The idea is to then collect data on all of these variables through 2049. So what type of data does NEON collect? So I mentioned that we have 180 different types of data, and we kind of categorize those into 3 different types. The first is going to be airborne remote sensing. So this picture that I'm showing on the screen is sort of an idealized NEON site, and you can see an airplane flying over. And essentially what happens is during the peak green time, so when the trees are all leafed out and the area is as green as possible, we schedule a flight over the entire site, and we have sensors on the airplane that includes things like LiDAR, hyperspectral imagery, and high-resolution photography. And we put all that data online and make it free and available to researchers. And we also have a large observational sampling component. And so you can see in the picture, there's a person on the ground that's kind of measuring the diameter of the tree. And we have people on the ground doing all different kinds of environmental measurements and taking samples from the organisms that we're collecting. And we measure a whole variety of different organisms as well through this observational sampling platform. And then finally, we have a really robust instrumented sensing system. And so one of the reasons NEON was started was to better understand climate change. And so sort of the iconic symbol that a lot of people think of when they know NEON, when they think of NEON sites, is this large tower that you can see on the left-hand side of the picture. And that tower has mounted sensors at different heights, and that allows people to look at flows and fluxes and different temperatures at different heights. And we also have sensors distributed throughout the site as well, looking at factors like temperature, soil temperature, and precipitation and vapor pressure and all kinds of things. And so the most important aspect of all of this is that the measurements are all collected in the same way across space and time. And that's again what allows us to do this continental scale monitoring. I wanted to show this slide because it kind of gives a different picture of what it is that we're collecting at NEON. So we have atmospheric data. We have data on a wide variety of organisms. That's the area that I'm involved with the most at NEON. And so that's what I'll be speaking to a little bit more, especially since I think it's most relevant to the introduction of mosquitoes and the types of things that are maybe most likely to be impacted by that. So we collect data on a variety of organisms, from plants to organisms like beetles, fish, birds, small mammals, mosquitoes, and ticks. And so that's our organismal sampling. And we look at all kinds of things from abundance and composition to biomass, productivity when we start getting into plant aspects, pathogens in some of the taxa, their phenology or seasonality. And then we also, for some of the taxa, are also looking at marker genes and metagenomics. Some of the other variables that we're also measuring are ecohydrology variables, factors that are related to biogeochemistry, and then the remote sensing aspects that I talked about. And so another unique aspect of NEON is just this really rich set of data and metadata that are being captured in the environment so that you can better understand some of the processes and drivers of ecological patterns that might be observed in the landscape across all these different sites. One of the other really exciting things about NEON is our biorepository. So in addition to the data that we're making available online free for researchers, we also collect samples from a lot of the organisms where the field folks are on the ground actually collecting those samples and then sending them off to the biorepository. So the biorepository is actually located at Arizona State University, and they're housing 65 different sample types. And so I have a whole list of those on the left. And as an example, for instance, the small mammals, we have samples including hair samples, blood samples, fecal samples, and so if an animal dies in the trap, we get vouchers. So we have a whole bunch of different sample types for each of these different categories. And what ends up happening is that the biorepository is receiving 100,000 specimens and samples per year, and putting all that information up on their website. And then those are requestable by universities to do all kinds of analyses that are beyond what we're capable of doing as an organization. So as an example, a researcher might be really interested in the chemical composition of the soils, looking at maybe the isotopes that are present in the hair samples from the mammals, or hormones, microorganisms that are present in the samples. And the biorepository really enables a lot of different types of research in that way. And so that's one of the really exciting parts of NEON. And so with that, I wanted to go into a little bit more detail about the terrestrial and aquatic sampling that we do, because those seem to be the types of sampling that would be most relevant to mosquito releases. So to start out with, for the terrestrial sampling across the 47 field sites, we hire about 250 seasonal field technicians, and they're collecting data using standardized protocols. And one of the ways that we ensure that the data are getting collected in the same way across space is a really robust training program. So they read all of our protocols and they take quizzes on those. And then we have certified trainers that work with them to make sure that they're collecting data according to the protocols. And then each year we have a handful of sites that are selected for auditing, where an auditor will go to the site and make sure that data are being collected in the same way. So there's a lot of quality control over the collections that are being done. And the types of data that we're interested in getting. So to begin with, on the organism front, most of the organisms we're sampling abundance and diversity. Those are kind of some of the key variables that people are interested in. So we do that for everything from plants to soil microbes, small mammals, mosquitoes, birds, ground beetles, and ticks. And so you can see a couple pictures of some of our different field technicians out in the field actually doing this. Is sort of a beetle sample cup. There's some folks obviously doing some plant work here. And then it looks like maybe a little bit of small mammal sampling in that other picture. So core variables being abundance and diversity. For a handful of taxa, we're also interested in the pathogens. So this would be small mammals, mosquitoes, and ticks. Small mammals started out, we were looking at hantavirus. That was of great interest when the observatory started. And then the researcher interest sort of petered off, and there were a couple of issues with that particular data set and a lot of requests for us to look at tick-borne diseases in the small mammals. So we were responsive to that, and we ended up changing, and now we do PCR testing for tick-borne pathogens in the small mammals as well as in the ticks. And so we're testing for the same suite of pathogens in both, and that really makes the data comparable. And we have this really neat disease ecology data set to analyze tick-borne pathogens in the ticks as well as the small mammals. And some of the pathogens we're looking for, of course, Borrelia burgdorferi, which is the pathogen that causes Lyme disease, but we're also looking at several other pathogens, such as those that cause ehrlichiosis, anaplasmosis, and many others. And then in the mosquitoes, we're using PCR to detect a range of mosquito-borne pathogens. So this, of course, includes West Nile virus. That's the one that we tend to see the most in our samples that we analyze, but we get a few hits for a variety of different encephalitis viruses as well. All of the viruses are exceedingly rare, and we're actually in the process of putting out a survey to get user feedback. And we may be rethinking the way we do our mosquito pathogen testing, perhaps looking at things like metagenomic sampling or other things. And so that is an area, if anyone's interested, I'm happy to pass along the survey link. It's going to be open till June 7th. So we have the rest of the week if anyone would like to fill that out. Finally, on the terrestrial side of things, we're looking at the biogeochemical and plant processes as well. So we measure below ground biomass of the plants that are under the ground, litter fall, canopy foliage, carbon and nitrogen pools that are in the soils, forest down wood when you've got trees in the areas, herbaceous biomass, and then leaf area index. So just giving you a hint of all the different kinds of samples and data that are being collected. This is just a different way of visualizing that. And this is sort of giving you a sense of the whole suite of what we're collecting and what data are available for researchers to sort of combine in unique ways to ask really interesting scientific questions about environmental patterns and processes. So on the upper, for each of the columns are the different taxa that we sample. So you can see that up on the columns, and then the rows are the different types of data we're collecting. So as I mentioned, all of these are having diversity and abundance measurements, pathogens at just a handful. So phenology or seasonality is being collected for a couple of the different data products. And, the fluxes, biogeochemistry, metabolism, and productivity are also a part of the terrestrial sampling. So with that, I'll move on to talk about the aquatic sampling that we're doing. We use standardized field techniques. And again, we have the same 250 seasonal technicians. A lot of them are housed in the same area, so they tend to be experts in a variety of different protocols. So it's not just one technician doing one protocol. They're really experts in a lot of the different types of things that we do. And we have standardized field techniques for all of the things that we're doing. As I mentioned, when we formed the observatory, we worked closely with partners that are also collecting long-term data. So folks at the USGS and EPA, LTER sites, to understand how they're collecting their data so that our data might be combined with that if someone were interested in expanding the spatial scope of some of the things that we're collecting beyond the sites that NEON actually has. And for aquatic sampling, we use a community approach. So we're sampling at each level of the aquatic community. So that includes the primary producers like periphyton and phytoplankton and aquatic plants, and then the consumers of those things. So that's zooplankton and the macroinvertebrates that we find in the water, and then moving on up to the consumers and predators, which is the fish. So those are some of the things that we're sampling. In addition to those biological samples, which are listed here, we're also looking at a lot of the physical characteristics of the water environment. So that includes bathymetry and morphology, as well as discharge, and then the geochemical aspects as well. So sediments and water chemistry. So again, just kind of giving you a broad spectrum scope of the types of things that we're measuring at NEON. And again, this is the same diagram, just looking at the aquatic aspects of what we measure. So fish, macroinvertebrates, microbes, algae, aquatic plants, sediment, and water. Again, looking at diversity and abundance as main focal aspects of variables that we're measuring, but also interested in metabolism, biomass, and pools and fluxes. So I wanted to just give a broad spectrum overview of the scope of NEON so that everyone has an understanding of what we do. And then next, I wanted to talk about, I think for this particular group, it might be a smaller area of focus. So I'll talk about how to find more information about the data that we collect, the protocols that we use for collection, and just information about how we came up with the designs in the first place. So that'll be sort of the next portion of the talk. The first place you'll want to go when you're interested in NEON science is to the data portal. So that's at data.neonscience.org. So that's the URL that's up here at the top of the screen. And this allows you to explore and download all of the data for free. And we have a lot of programmatic access as well, and a lot of tutorials and things for those that may not be as familiar with working through APIs and things like that for how to access the data. We also have a lot of code packages developed to make the whole process a lot easier. And it's all documented on the website. If you're interested, which I suppose most of the folks on this call would be in the data product protocols and the documents associated with how we do the design of the science around this, the first place you're going to want to go is under the Explore Data Products part. So it's this left-hand upper section of the website. You'll want to hit the Explore button. So when you do that, I have a picture on the next slide of what you end up with. You end up with a search screen here on the left side, and you can type whatever information you're interested in. For this particular group, I typed in mosquito into the search bar here, and you'll end up with all of the different data products that NEON collects that have the word mosquito in the title and possibly also the abstract. And the first thing that came up for me was the mosquito pathogen status, which is helpful because that tends to be what folks are interested in here. So if you click on that, first, I suppose I'll show you how this timeline here at the bottom is showing you how much data is available. So you can see that we go, from 2018 through 2022. And actually there's data from before that time period as well. But all the dark colored boxes show you how much data is available for different time points. And you can see getting into 2023, the release data is not yet available for these. And so if you click on the link up here for mosquito pathogen status, that will take you to a page that gives you a lot of details about the mosquito pathogen data product. Is this a question?
No, go ahead, Sarah. I think somebody has their microphone.
Oh,
open,
okay,
so
gotcha. Okay.
if people could mute their microphones. That's great.
Okay, thanks. Great. And so it'll take you to a data product page where it tells you all about how the mosquitoes are sampled and collected. It'll give you a little bit of information about how long it takes between sample collection and the time at which it's available on our data portal. And then if you continue scrolling down the page, you get, again, a little bit more description. And I think the part folks are going to be most interested in is the documentation. And so there's always going to be a standard operating protocol. So that's the really detailed, long description of exactly how everything's done. In this case, it's a lab protocol because this is the lab that's analyzing the samples for pathogens. There's also going to be user guides. So there's a quick start guide for those that just want to get started with the data and want the quicker version. There's also a more detailed user guide. And then the scientific design and rationale is also going to be on here. So for those that might be interested in understanding how we came about making decisions about which mosquito pathogens to monitor for and how to monitor for mosquito abundance and all that, those are all going to be on this page as well. So scrolling through the documentation is really going to give you a lot of information about that kind of thing. And so next, I just wanted to give a little bit of history about why we're measuring the things we're measuring and how we made those decisions. And I think hopefully that will be helpful for folks that might be interested in forming their own types of monitoring or observatory types of products. So NEON's goal, the stated goal, is to improve understanding and forecasting of ecological changes at continental scales. And so some of the things that we're really interested in are climate change, land use change, and invasive species, and how those can be observed at a continental scale. And NEON our mission is really to provide infrastructure that supports not only basic research, but also education and environmental management in these areas. And so one of the questions is, how did we come up with the different things, the different areas and topics of focus? And the answer is that we were really focused on the grand challenges in environmental science. So those were outlined in 2001 by the National Research Council as high priority environmental science topics. And you can see on the left side here what those topics are. So they include biodiversity, biogeochemical cycles, climate change, ecohydrology, infectious disease, invasive species, and land use. So the goal here was really to design an observatory that would give data that would allow folks to make really novel and transformative discoveries in all of these different areas. So that's one of the reasons why we're measuring so many different things at NEON. And so how did the observatory go from this concept of the things we want to measure and make available to people into what it is today, which is pumping out, millions and millions of lines of data for folks to analyze on, an annual basis. So NEON was conceived, as I mentioned, in the late 20th century as an instrument to advance the ability of science to examine interactions between life and the environment at the continental scale. And so from 2000 to about 2006, there was a series of workshops about building this comprehensive ecological observatory network. And the initial plan was completed in 2006 and submitted to Congress for funding. And then, of course, that took 5 years of back and forth about revising the design and making it, as good as it could possibly be. And then Congress approved the funding in 2011. So that's kind of when construction began, was around 2011. But of course, we're constructing sites all over the continental US, in variety of complicated ways, towers that are going up, and we need to think about all kinds of site access, permitting, all the kinds of things that you need to think about. So construction of all those sites was completed in 2019, and NEON entered the operations phase in the summer of 2019. And then again, of course, that's going to be for 30 years. So it's planned to continue through 2049. And the planning was very extensive. So we did a lot of collaboration with agencies, as I mentioned, to ensure that the data are comparable. We also continue consulting today with technical working groups, and they also played a role in the development of the protocols. So a technical working group is just a group of sort of 6 to 8 subject matter experts in a particular area where we collect data, and we can ask them if we end up with questions about sampling or issues that come up during sampling to make sure that we're doing things in the best way possible. And then, of course, during that construction period, there was a lot of prototyping and working with equipment and sensors to make sure that it was going to be fully online and functional by 2019 when we started. And we had to incorporate feedback from field teams when things weren't working quite as the way they were planned. So as I mentioned, in terms of the spatiotemporal design, I sort of mentioned how we grouped the continent into different ecoclimatic domains. Within each of those domains is a core site. And one of the things we thought about when we selected those is making sure it was in a wildland area. So it was a good kind of baseline measurement. It had to be representative of the domain that we were looking at. And then we had to think about permitting and having access to the land for 30 years. And then within the gradient sites, we wanted to enable questions about these grand challenge topics. And so we were using those gradient sites in strategically located areas so that we could compare them to the wildland sites to get an understanding of things like agricultural systems that might be different across the country, or different forest management histories, areas that might be experiencing different levels of invasion, or different types of atmospheric transport that can occur in sort of mountainous regions and things like that. So that is how we selected the sites and the spatiotemporal design. And then the next question was, what organisms are we going to monitor? What are we going to be sampling and why? And so one of the things that we really focused on was developing sentinel organisms that would allow us to look at things with a variety of different life histories. So for instance, we're looking at soil and aquatic microbes. Those things are going to live for about a day. We also look at algae, zooplankton, aquatic invertebrates. Those things live from weeks to months. And then we move up into things that are more long-lived. They live for years. So these would be things like beetles, fish, small mammals, birds. And then, of course, terrestrial plants are going to span the entire gamut of all of that. And this allows us to look at things with variable lifespans and how different changes like climate change might be influencing those in different ways. And then also looking at things with widespread distribution. So we, of course, want to be able to enable those continental scale comparisons. So I really wanted to finish up by talking about some of the successes we've had, as well as the lessons we've learned along the way, in the hopes that this can help folks start out with maybe a strong foundation that might be interested in developing some of these observational types of facilities. So in terms of developing the field sites, some of the things we had to keep in mind were making sure the size was reasonable. So that would allow us to support access points, roads and trails, and of course utilities, and access to electricity and things like Wi-Fi are really important, especially for our sensor networks. And then on the other side of the equation is thinking about the relationships that we're building. So investing in building local relationships with landowners and surrounding residents has been really important for us. And that just ensures that the local community knows what we're doing and perhaps is benefiting from it as well. So I know we have a lot of really neat examples of that kind of thing at NEON, and I'm happy to talk more about those. I think I'm running out of time to go into great detail there. But those local relationships are really important, and co-benefits is good for just ensuring that the site access is maintained throughout the duration of the study. Flexibility was really critical. So the field instrumentation required a lot of prototyping and new designs. Sometimes things become obsolete. One of the things that happened was, these sites would have these major floods, and we have stream instrumentation in there designed to measure water flow, and then it would get dislodged during the flood. And unfortunately, the flood is the thing we're interested in, but the thing got dislodged during the flood. So we had to go through and design different ways of mounting these things so that the data would be comparable, but we could still collect data during these high flow events. And so just kind of some interesting examples of things that we had to shift up, make sure the data were still comparable, but had to be flexible at the same time. So if you're interested in more examples from these, there's a nice paper that came out last year looking at lessons from constructing and operating NEON. So I have the citation there at the bottom of the screen if you want to look at some more of examples of those. Of course, community engagement is one of the key aspects of NEON, so that's kind of why we're here. We engage early and often. We want to make sure that people feel like they co-own all of this data. We're very transparent about some of the problems that arise, issues, data gaps, things that come up. We have a data notification page and lots of other ways of notifying folks that might be using the data of discrepancies and things they might want to be aware of. So we want to make sure that we're proactive about things that come up and letting people know when that happens. And then another thing is just to leverage the partnerships we have. So we've developed really strong partnerships in a wide variety of areas to make sure that we're not duplicating efforts, right? So if the community has come up with an algorithm that helps them use our data, we might work with them to incorporate that algorithm into a thing that becomes more visible on our webpage. We do a lot of data quality reviews. We work with the NEON partners to look at things like training and internships for the educational portion of what we do. We share our data with other organizations that might host our data for free. And so we're getting visibility through their website, and then they're getting additional data for their website. So it's kind of a win-win situation there. We do a lot of facilitating research and education. We work with organizations that help NEON connect with underrepresented communities. And so we really leverage these partnerships in a really meaningful way to get as much as we can out of what we're investing in NEON. In terms of cost planning, it's really important to think about the fact that construction is never fully complete. So infrastructure gets damaged. We have to maintain the software. If we have flat budgets, things are going to end up having to be dropped over time, especially if it's a longer term monitoring plan. Because of things like inflation, labor and materials, all those kinds of things are expensive and they continue to grow in cost. And so planning for the things like sensors becoming obsolete, needing to buy new things is going to be a really important aspect of budgeting a large scale observatory infrastructure like this one. So those are the lessons that we learned, but I also wanted to talk about some of the successes that NEON has had. So the graph on the left is just looking at the number of publications that have come out. And to date, I think we're pushing 600 publications that use NEON and, tens of thousands of citations of works that have NEON data or samples that are incorporated into them. And then, of course, it's not just a US-based, all of the data collections are occurring in the US, but it's a global enterprise where folks are downloading the data from all over the world and using it to answer really interesting ecological questions. The other nice thing about NEON is that we're there before a disturbance happens as well as after. And so we have a lot of really great data for baselines for change. So this can be major ecosystem disturbances like fires, hurricanes, droughts, and agriculture. And so there's a couple different papers looking at different examples of fires that happened, hurricane winds that blew down a lot of trees, and agricultural changes at sites where cattle were brought in. So lots of potential for different types of questions. And I wanted to just end with a grand vision that we at NEON have, that we have basically just a global network of networks. So we'd love to see other networks pop up like this, where free and open and standardized data are being collected to understand and address some of the world's biggest challenges. And so that's all I have today. I did want to thank the organizers for inviting me to share, and I'll let them take over.
Thank you very much, Sarah. Thanks a lot. Very interesting. And let me encourage the audience to, well, first of all, let me say to the panelists, thank you all for presentations. If you could all turn on the panelists could turn on our speakers could turn on their microphones and their cameras. We have some time for questions, and let me encourage the audience to chat in questions, or you can raise your hand and we will try to unmute your microphone. You can ask your question or make a comment orally. We do have some questions that have been coming in during the course of the presentations, and I'm going to take a few of those now. And but I certainly encourage other people to continue to ask questions. Paul, we'll start with you, since it's been a little while since we heard from you, and we'll just sort of get refreshed on your topic. But one question that would be interesting to hear your response to in particular is what response would be expected if post-release monitoring showed an adverse effect on a non-target organism?
Yeah, this is an interesting question because we actually don't have a lot of recent precedent for this, because most of the adverse non-target effects that have been detected are from releases before the 1970s. However, we can look at some of the guidelines from places like NAPO and some of the documentation in Europe to know what would be expected. And it would be obviously context dependent, but the first thing that would happen for sure is that releases would be discontinued. So for example, if after 1 year of releases in Italy, it spelled out pretty clearly that if there were significant non-target effects, that subsequent releases would not be authorized. And then depending on sort of the specifics of the system and how feasible eradication would be, sometimes it's just cage releases that are authorized at first rather than open field releases. So sometimes cage releases that are looking for non-target impacts, if they did, basically that would be destroyed and sprayed with insecticide. If it's an organism that's introduced in a small area and doesn't have very good dispersal, the expectation would be that it would be just sprayed. I know of one past example in weed biological control where a recently established population was intentionally eradicated with insecticides after initial results. That was quite a bit in the past, in the early 90s, but for these retrospective evaluations of releases that happened 30 years ago, where the organism's already widespread, I guess an example is the cane toad, where there's a huge conservation and management response in Australia for an introduction that happened in the 1920s. But yeah.
Okay, great, thanks. Renee, a couple questions that have come in. A couple technical ones. One is, do you have an idea of the FST values of the Hawaiian mosquitoes compared to the mainland mosquito populations? And is there any hope of locally fixed alleles that a gene drive could target? So somebody thinking about possible applications of gene drive in your context.
Yeah, I don't have any data from mainland mosquitoes. That's kind of phase 2 of the project as it's ongoing. I do have hope for identifying local alleles here in Hawaii that might be good gene drive targets or other targets for gene drive or other forms of synthetic biology techniques. And with regards to, let's see. So yeah, hope for locally fixed alleles. Even locally fixed alleles island by island is a potential, just given that the structure was somewhat robust, especially across chromosomes 2 and chromosomes 3, not so much chromosome 1.
I had a question of clarification, I guess, from with regards to the mosquitoes that are being released. Are those naturally infected Culex, or were those infected with a form of Wolbachia that is known to be incompatible with the native the invasive species?
They're transinfected with Wolbachia that was isolated from Aedes albopictus supergroup B.
Yeah. Okay.
Yep.
Okay. So that was done in the lab, and so the infection here is one that isn't found typically in that species of Culex.
Correct, not in that
Yeah.
species of Culex, but Culex, unlike say Aedes aegypti, does carry Wolbachia naturally, and it has a really complex crossing type, so like 5 different crossing groups for CI.
Yeah, good, thanks. Okay, Jafet, a couple questions for you. One question was, is there a minimum data set size for which your correction models are valid?
Yeah, so well, in my experience, overall these approaches work well, even with smaller sample sizes, about 100 of locations, 100 of discrete sites. Obviously, you're a bit constrained by the resolution of your spatial information as well, and how complex the model you want to be. Usually for spatial models, explicit spatial model, you tend to need a lot of information in multiple locations. So try models with maybe 100 to 100 sites, and that's about the sample size where you're still able to estimate some of these parameters. In the spatial context, obviously, depends if you have a very large spatial temporal data set and the records are very sparse over space and time, you might need to end up aggregating your data over space if you don't have enough data points on a spatial temporal framework. But yeah, usually you're a bit restricted about the sample size being less than 200 sites. It's not really worth it to fit such complex models in a spatial temporal framework.
Yeah. Could you say something about how these models are being used or potentially going to be used, and by whom?
So it mostly a population ecologists and are the ones that are going to be using some of these models. Right now, we are actually working with an extension to these models for invasive species, because mostly one of the drawbacks of such a spatial temporal structure effect, it's that you assume that the spatial range of the correlation between your spatial structures remains pretty much this constant through time, and where species and they do not reflect this spread behavior of invasive species. So we're using some, extending this to incorporate some physical models such as these heat equation models, these differential equations to model the behavior of the invasive species to kind of be able to capture all the information from where you have earliest stages of when the species is introduced, and how can you monitor that through different time points. So far, our models are not really suitable for such behaviors. You will need data that it's similar in across space and time in terms of the volume of data. So we're trying to expand this now to actually be able to use it for invasive species as well. But mostly it's for population ecologists. We can also, for people in community ecology, it's also being used for aggregating the counts for multiple species. So that also, it's another possible of extending this. But these are mostly how I think most people are going to be using such models, or actually are using them at the moment.
Yeah. And just one more follow-up, I guess, for you. Say something about model selection. So, for example, when you were describing the catbird data, you showed three different models, or three different forms of a model, or that you showed. And so, how would you make a choice as to which is, quote, the best or the most useful?
Right. So for this, we use something that's called a leaf group out cross-validation. So basically, it's a new development of INLA in which you basically remove certain data points, and then you feed the model on all the remaining data points, and then predict that group you removed, and measure the difference between your predictions and the true values of that data. And so you do that for every single groups that you have defined. We have a way of constructing those groups automatically, or you can construct them by hand based on your expertise. The idea is basically to resemble the spatial structure or temporal in your dataset, and therefore you want to have the model that has the highest predictive accuracy. And so based on that, you will select that model, the one that has the highest predictive accuracy based on that metric that we've computed. So that's how we are using cross-validation for these sort of occupancy models. For the point process, however, it's difficult. Actually, there are no tools for doing model comparisons at the moment, because your data point, your data are actually the locations where species occur, rather than a discrete area that you sample. Your data are the points, and therefore it's also important to look where you don't have information as well. So basically, it's kind of like interpolation over space. And so all the metrics that people usually use, like information criteria or metrics that depend on sample sizes, are not really valid for point processes. So that's something that it's not only us that we're working on. It's on a general scope of point process models. How can you do some model comparison for such? So that's a much more tricky question that some colleagues of us are working on. But for the other models, we have this tool that allows you to compare the different models.
Yeah, good, thanks. Sarah, there was a question about which pathogens that you're looking for. Is there a list of pathogens that you look for in mosquitoes, in this case?
Yep, it's a great question. There is a list. So currently we're using PCR testing. The way we do it is essentially we start out by screening, because they're so rare. We actually pool the mosquitoes into groups of about 50, and then we screen them for larger virus families. So that's the alpha viruses, the flaviviruses, and the orthobunia viruses. And if any one of those pools comes up positive, we then go on and do a testing for individual viruses, and there's a lot. Probably the most recognizable ones are going to be West Nile virus. We do lacrosse virus, Japanese and St. Louis encephalitis viruses, California encephalitis virus. So those are the ones I think we've found. But we also look for, chikungunya, Orapuche, lacrosse, Zika, dengue. All those we're less likely to find them, obviously, given where we're looking, but they're a part of the multiplex assays. And so they work out that way. However, because of the rarity of the viruses, we're actually thinking about moving to a more metagenomics type approach, where we would be able to kind of put out the sequences that are found in the mosquitoes that we're analyzing, and then folks could use their own bioinformatics pipelines to find anything that's there, rather than a more targeted PCR-based approach. So if anyone's interested in that, I'm happy to share a link to the survey where we're getting feedback on that potential change.
I had a question on the auditing process that you folks implement to try to maintain quality control, and how successful that is, and how much variability and issues have you found with trying to maintain a very large collection of technicians trying to collect data for you in a way that's going to be useful?
Yeah.
and
Yeah, it's a
comparable?
great question. I think because of the amount of time that we've had to develop the protocols and develop the training materials around them, and because of the auditing procedures, things are really done, detail to detail. It's the culture, I guess, at the organization as well, is that if a detail's not going to be followed, it needs to be, put to the head scientist that's in charge of that particular protocol, to ask about the impacts and things like that. And so it is a cultural thing in terms of making sure that everyone understands the importance of doing it the same way. The training really emphasizes a lot. So as each year, we'll maybe come up with specific questions that come up commonly, and those get incorporated into the trainings that the folks are watching before they do their sampling. And then the auditing procedure has a whole list of questions that come directly from the protocol, and a yes, no of whether they've been followed, when the auditor comes out. And it's always extremely minor things that, something about the wrong kind of gloves being worn to do dry ice. You know what I mean? It's like a very detailed level of accuracy in terms of similarity of collection. So I think we feel quite good about that, and happy to talk about the process that was used to.
So the hundreds of technicians that you mentioned are strictly sample collectors, or are those samples then sent to a hub facility or a main facility where they're processed, where the quality of at that point may be more better controlled in terms of, say, molecular analyses and things like that?
Yes, that's a great question. So most of the things that NEON does is the on-the-ground field sampling and collection. And then we tend to have taxonomists identifying all of the mosquitoes and the ticks. So these are professional taxonomists, and the quality is highly checked at that stage. The beetles, I think we actually do a lot of that in-house, although there is confirmation that some of them are sent to professional taxonomists. So, and then obviously all the pathogen testing is done at an external lab. So there's a lot of contracts involved as well, in terms of how we manage things, and quality control at those stages as well, and auditing.
Yeah. Okay. Great. Let me encourage the audience to continue to chat in questions if you can, or continue to do that. Renee, I wanted to come back to you regarding non-target of impacts, which was a topic that Paul brought up during his presentation. And did you implement any kind of monitoring of non-target impacts of Wolbachia releases in your project?
We have not modeled them because the mosquito is not native to here. Then the target impacts of population suppression should be minimal to none in terms of how that would impact the other organisms that are present on the landscape. The mosquitoes are not believed to be a major food source for any animal. And so with that respect, the only kind of impact that we could envision potentially would be, they do feed on nectar. And so there's going to be a little bit of resource use, and potentially there would be potential for pollinator behavior, but because they're not native, that really shouldn't be a negative impact. There's the impact of the helicopters as a noise. And so that's not really well, it is an environmental impact, but not an ecological impact per se.
Okay, good. I guess so Paul brought up the ideas of food webs and ecologists, and being aware of food webs in order to facilitate detection of indirect effects. And so in the case of the Culex mosquitoes, I mean, is there a place within the food webs in those environments known, or
We have looked at,
that was to you, Renee?
thank you. We've looked at bats, for example. Our only native mammal is the Hawaiian hoary bat, and mosquitoes do not comprise very much of their diet at all. In terms of birds, we are looking at at the bird samples, but the bird samples seem to eat a lot of moths and not, we haven't looked for mosquitoes specifically in their diets, but that's something that would potentially be of interest. But again, because the mosquitoes overarchingly have a negative impact relative to any kind of impact that might have on by removing them on the food web, that would be very small relative to the impact that they're having on the birds, which is basically taking out the pollinators and these keystone species for the forests.
Yeah, interesting. So in thinking about Jaffet's talk about sampling bias and availability biases and things like that in sampling schemes, I was wondering, again, Renee, I was thinking about the rugged landscape that you had described and showed in some of your pictures and how that impacts sampling schemes in this case, and whether or not, well, if the program envisions any sampling bias as a result of some of those constraints.
Yes, there will be a sampling bias because of access to the different sites. And so at present, well, last year in 2022 and 2023, the USGS went in twice per month into that area in the Kipahulu Valley. And so we had standardized data collection for an entire year and altered each site. So it's basically one site per month to look at mosquito abundance and seasonality, do some monitoring of the landscape. But the access is very difficult and it's expensive. And so that is going to be problematic. As a component of our IIT program, there are requirements to do some types of monitoring, in particular the oviposition cups. And then we're running adult mosquito traps as well. And by we, I mean the groups on the ground, Maui Forest Bird Recovery Project, and soon to be the Kauai Forest Bird Recovery Project. And so they have transects that they're implementing the adult trapping efforts on. And then they're collecting the mosquitoes in the traps. And then those mosquitoes, some will be sent to USGS for looking at disease suppression, and some will be genotyped to look for a response to the IIT treatment. But that doesn't get at the sampling bias question, which is it's a hard one.
Yeah. Is the program that you're involved in right now sort of in testing phase, or is it really now at a point where it's full-blown implementation with the intent of full coverage of the range of the species, the target species?
I don't want to define it, but I would say testing if I had to, because it is a rather constricted area and it's not range wide across the entire island. And so it's basically a stopgap measure to prevent the birds that are there from going extinct in this very narrow time frame that is becoming increasingly shorter because the birds are the populations are diminishing faster than had originally been anticipated. And so as a stopgap, this is something that we can do to try and prevent extinction, but we need more durable tools ultimately.
Yeah. Yeah. Paul, Gene Convene was running some webinars over the last couple of years, and we had a speaker on, and I can't for the life of me remember his name, but he was a plant, worked on invasive species of plants, and actually wrote an interesting paper. And of course, I'm blanking on his name, so I can't tell you who it was. But I guess the interesting thing that he found in looking was indirect effects have had major consequences on. I guess he was looking at control programs, or maybe it was the effects of invasive species themselves in terms of their effects in the environment. And its point was that indirect effects were more significant than direct effects. And I think, as you said, those are the hardest things to measure. And it's because it's hard to figure out what to look at, because there could be so many indirect effects. Anyway,
Yeah, that's right. I think what we've
talk a little bit more about these indirect effects, and you talked about trying to get more ecologists, perhaps involved in these programs. And so maybe you could say a little bit more about that, how that might they would really enable enable you to do a better job at what you guys do?
Yeah, I think something we've learned about both direct and indirect effects from these biological control systems is that the sort of relative importance of direct and indirect effects is very case dependent. Every every food web is really different. I think, as I mentioned during my talk, the sort of long-standing philosophy recently, I guess, has been if we can minimize the amount of direct effects, that'll minimize the potential for indirect effects because they tend to sort of flow from direct effects. But then, of course, I presented a case where that's not the case too. So it's kind of been indirect effects has been a kind of a learning process because we have a lot of modeling on indirect effects. There's all types of modeling you can do on theoretical food webs to look at what the potential indirect effects can be. But the sort of challenge about these biological control or gene drive systems is that you kind of need to, it's where the rubber hits the road, and you're actually faced with having to try to predict or evaluate them in real systems. And that's been really challenging. So it's one of the most active areas of research in biological control right now. But because in practice, we've only observed really one type of indirect effect in reality in the wild, which is this we call it an enrichment effect, where the biocontrol agent's ineffective and gets really, really abundant and acts as food for other things that spill over onto other species. We'd almost need to measure more cases of where biological control has gone wrong and caused indirect effects. And obviously we can learn from invasive species research there too, but to then know what to look for in the future. So the advantage, I guess, that we have is that we have some history to look back on when you're considering a new technology like gene drives. There isn't that sort of empirical track record to fall back on. So that's definitely a challenge.
You mentioned modeling. Is the biological control community relying more now on, would it be ecological monitoring? What kind of modeling, I should say? What types of, how is modeling fitting into current biological control implementation?
So biological control and modeling have like a long history together. In fact, a lot of the first population dynamics models, like the Nicholson-Bailey predator-prey models, were sort of developed from post-parasitoid biological control systems. There was a ton of work in the 80s about theoretical using models to understand sort of the theory behind biological control systems. But there's a lot of angst in this biological control research community that modeling has not really served a practical purpose yet for informing biological control introductions. Models have produced useful heuristics for us about sort of guidelines about what to expect. For example, if we see a certain level of non-target attack, how likely is that to translate to a significant population level impact? So we have like heuristics, but in terms of actually using it to be predictive about what we might observe in terms of non-target effects, it hasn't paid off yet. So modeling is used right now to understand sort of the fundamental processes that underlie biological control systems and their ecology, but it hasn't yet, in my opinion, and the opinion of sort of a lot of my colleagues who are applied biological control practitioners, it hasn't paid off in terms of sort of directly directing the programs.
Yeah, good, thanks. Lots of discussion in the gene drive community, certainly lots of models being made, and the question is how useful they'll be and how they would be used by decision makers down the road, for sure.
My perspective is that they're useful for thinking about how the systems work and what factors are important and the relative importance of different factors. And that's sort of how we think of them in biological control. Is they're useful for making our assumptions about how these complex systems work explicit and thinking about the different components and how they affect each other.
Yeah. Good. I see we're right at our stopping point. So at this point, let me reiterate my thanks to the speakers today. Thanks for a set of very, very interesting talks that related to each other, and I think were useful in generating some thoughts within the audience as well. And so really, really interesting. And thanks again for doing it. I'd like to thank the audience for your participation. And I think with that, we'll say thank you to all, and I'll end our program and say have a nice day.